Hello, good evening, everyone. Hello, very good evening, all of you. Hello, very good evening, good evening, all of you. Hi, this is Vaishali Kamdar from Spark Academy, and today we are going to discuss about cell cycle and cell division. Cell cycle and cell division. So, before starting the topic, let me remind you all of you that you can follow us on any of these links, any of this thing, that is sparkacademyonline.com, Instagram, Telegram, Facebook. You can follow us, Spark Academy Online. So what are you all waiting for? Or else you can download our app, that is Spark Academy app from Play Store. Then you will be having access that which all courses are going on. If you are still worried, it's high time now to join the classroom coaching or online coaching. So short term regular courses have been already started since long. Now it's time to start crash course. So classroom crash course coaching has been started since 1 week, but you can still join it. Still join it. You can, if you are not willing to join classroom coaching, then you can join online coaching also. See, on daily basis, we are taking this, conducting this live classes, daily live classes. So that is on YouTube, our YouTube channel, Spark Academy channel. So you can subscribe to our channel, and you will be having all the access to the live classes which all are going on daily basis. These all are mostly one shot program. So we are discussing all the topics required for NEET, JEE, EAMCET, etc. So what are you all waiting for? You can go and search Spark Academy. Don't forget to look into the icon, that is the logo. This is Spark Academy logo. Okay. So this is a complete team. This is a complete team, and this is me. That is Vaishali Kamdar, and I'm your biology teacher. So this is a complete, a team of NEET, JEE, and EAMCET. So this is a schedule for YouTube classes on daily basis. So you can have screenshot of it. So you will not miss out any of these classes. Monday and Wednesday, 5 to 6, it's my time. And those who are, for MPC students, maths class also will be there, the same timing. So this is Vaishali, and I'm your biology teacher. So you can take the screenshot. Come on, guys, take a screenshot and don't miss out any of the lecture. So when you want to join any of the courses, you can apply this coupon code. You can apply this coupon code here. That is VAI10, and you will be getting great discount. So great discounts. So to get discounts, you need to apply this code. So what are you all waiting for? This is high time to join the classes. So uh, join any of the course using this code. and you will be getting great discount. So let's start our today's topic. So let me tell you about the weightage part of the chapter. So it's cell cycle and cell division. As I've told you before that this is very sweet and short chapter containing only 3 chapters. The cell which you are going to, this is you are learning from 8th standard. Biomolecules is common for chemistry and botany. So chemistry and biology, it's not common you can say, but it is there in chemistry also, it is there in biology also. Then this is cell cycle and cell division, which gives you around 2 questions. So 2 questions, that means around 8 marks. Very short unit and still it will give these many marks. Then why should we not clear all our doubts of these chapters, these units? So I'm here to solve all your doubts. So you can join it live. You can ask me in form of comments also afterwards when you are watching the video. So I'm there to answer you. So whatever may be your doubt, you can post it in form of comments also and join it right now. Join it right now. You can join live sessions to discuss it more thoroughly. So I can explain it more thoroughly during live classes. So now we are starting cell cycle. See what exactly cell cycle means. A cycle, it's a life cycle of the cell. So during this life cycle, cell grow, perform its all the functions, replicate and divide. Again it will enter to the growth, functions, replication and divide. So this is how for eukaryotic cells, cell cycle consists of 2 general phases, interphase and mitotic phase. So cell division is a part of a life cycle for almost every cell. So it is most complicated process in eukaryotic than prokaryotic. Eukaryotes have multiple chromosomes and many organelles. So all of which must be duplicated and separated for the cell to divide. If I will give, during eukaryotic cell division, if a cell thinks that only I will give chloroplast, but I will not share my mitochondria. It should not happen like that, right? All the cells, all organelles should be duplicated and it should be given away in equal amount. It should be given away in equal amount of both the cells which have been newly formed, newly synthesized. So that is what we want. So sequence of event by which a cell duplicates its genome and synthesis other cell constituent and eventually divides into 2 daughter cells is known as cell cycle. That means starting from a single cell, chotu cell, right now it has been born, it will undergo growth. Okay, then again it is dividing. This is bigger cell, now it becomes grown, and now eventually it is dividing to produce 2 new cells. Okay, and all of that cell again re-enters into the same cycle. So this is what a cell cycle is. Starting from a cell to the cell division, 2 new daughter cells. It is known as cell cycle. So cell cycle is divided into 2 different phases, interphase and M-phase. That means preparative phase and you can say actual division. See, when you want to cook anything, difficult recipe, say for example, I want to make a brownie. Okay, so in that brownies, for making brownies, whatever is my requirement, first I need to collect all this, gather all these requirements. Okay, sometimes if I want to make some different recipe, then I need to chop the vegetables and all these things. So that is what a preparation time. Okay, directly we can't make brownie. That you can switch on the OTG, switch on the oven, and you can make it. So it should be not like that. For that you require some preparation also. So actually this interphase is a preparative phase. So normally we used to say that interphase in that cell is not active, then it is wrong. Cell is active. Cell is active here. Cell is active here, and it is its preparative phase. It's just that it is the phase where cell is not dividing. Okay, so in this preparative phase, what all preparation cell need to do? According to that, interphase is divided into G1 phase, S phase and G2 phase. That is between two synthesis part, there is one gap. Okay, that means, and then which will the next it is from M phase, which is karyokinesis, which is divided into karyokinesis and cytokinesis. So nuclear division and cytoplasmic division. So caryon, that means nucleus. So nuclear division and cytoplasmic division. Nuclear division is further divided into 4 different phases: pro, meta, ana and telophase. I hope this much is clear, guys. So now we are starting from the preparative phase. Okay, we want to make brownies, right? So brownies we want to make. For that, what all things you will be requiring, that things will be, we will be gathering all these things. If I need to buy it from a shop, I will go there and I'll buy that. So all these things I require. So this preparation for, see, this is preparative phase for cell division. So what all things will be required for cell division? See, I have told you previously that during cell division, you need to give away all the organelles, all the organelles in equal quantity to both the cells. Okay, that doesn't mean a cell will give away its own organelles, then cell will die. Okay, so it will synthesize, it will duplicate new to make it new. So first step is interphase. So interval between two successive cell division is termed as interphase. Cell division and the next cell division between this whatever interval is there, it is called interphase. That's why the name itself saying interphase. Interphase involves a series of changes that prepare the cell for the division. See, what is it? It's a preparative phase. So it is a period during which cell experiences growth, DNA replication in an orderly manner. So interphase is the longest period in which a cell cycle occupies more than 95% of the duration of the cell cycle. It is divided into 3 phases: G1, S and G2 phase. So this is how it will be. So this is G1 phase. So in here it is in G1 phase. One second. So in G1 cell starts dividing, it will duplicate all, it will make whatever things required for DNA replication. Enzymes, proteins, all these things required for DNA replication is synthesized in G1 phase. Okay, if any of the cell doesn't want to divide further, then it will enter into G0 phase or GO phase, that is G quotient phase. Okay, otherwise it need to proceed for the DNA replication. If DNA has been replicated, then there is no comeback. Then cell needs to divide. It is compulsory for the cell to divide. So in S phase, it is also known as synthesis phase. So what is synthesized here? DNA. DNA is synthesized by DNA replication. DNA replication occurs here. Histone proteins are also synthesized in this phase. Then G2 phase, here the duplicates chromosomes for the error and making indeed repair. So repair is done here and all the organelles will duplicate over here also. So first G1 phase. So it's the first phase of the growth. It is the stage during which the cell grows and prepare its DNA for replication. So it is the phase the cell is metabolically active. Remember, I have told you cell is active during interphase. So a lot of protein and RNA are synthesized during this phase. Proteins and RNA are synthesized during this phase. Large number of nucleotides, amino acids. So nucleotides which synthesis which is there in synthesis of required in RNA synthesis as well as DNA synthesis, and amino acid for the synthesis of protein. For histone synthesis and energy rich compounds are formed. Then in S phase, it is stage during which DNA synthesis occurs. So in this phase, the amount of DNA doubles. Amount, for example, it is 2C amount, then it gets 4C in S phase here. That doesn't mean number of chromosomes get doubled. That doesn't mean we have 46 chromosomes, instead it gets doubled. Okay, number of chromosome remains same, only amount of DNA per cell doubles here. But the chromosome number remains the same, and each chromosome now consists of two sister chromatids. Previously it was, before this point it was like this. Only one chromatid is there. Now for each chromatid it is replicating and it will form sister chromatid. So that it results into formation of a chromosome. Okay. So sister chromatids are held together by the structure called, this is called centromere. Now cell is not committed to, the cell is now committed to division. Okay, it is compulsory for the cell to, it is commitment for the cell to divide. Sorry, it's very hot, so we need to be hydrated when we are speaking to for a long time. Next, it is G2 phase. So in this phase, cell continues for the growth and prepare itself for the division. So proteins and RNA required for the mitosis they are synthesized here. Okay, remember proteins and RNA required for DNA replication are synthesized in gap 1 or G1 phase. In G2 phase, the RNA and protein required for the mitosis are synthesized during this stage. Then G0 or quiescent phase. So it is the stage where cells remain metabolically active but do not proliferate unless called to do so. So such cells are used for replacing the cells lost during injury. So this is G0 phase. So G0 phase or quiescent phase. So non-dividing differentiated state. Most human cells in the G0 phase, like liver cells in G0, but can be called back to the cell cycle by external clues. The nerves and muscle cells, highly specialized cells, they are arrested in G0 phase. They can never divide further. Okay, whenever any nerve cell is dead, it's dead forever. So it will never divide. Liver cells can go back. They can call back again whenever there is external clues. I hope this is very important, guys. So I hope you are noting all these points. Very, very important point from this chapter to note down about liver cells and nerve cell and muscle cell. So these are, they have called back, they can be called back. These cells, they are arrested here, arrested in G0. Then phases of mitosis. See, we have completed cell cycle, in that we have completed this interphase, longest phase. Now we need to move towards a mitosis, that is M phase. So phases of mitosis. Mitosis is divided into 4 stages: prophase, metaphase, anaphase, telophase. Cytokinesis completes the mitotic phase. Okay, see, let me explain you. So first it is first when after interphase there is a cell will enter into prophase. So prophase is divided, it is early interphase and then prophase, then it will enter into the prophase. Okay, so here you can see prophase. Wait a second, this is a prophase. Okay, this is early interphase, then late interphase. Now it is entering to the prophase. So prophase, here ester or microtubules formations are there, and the cell, this nuclear membrane gets disintegrated. Here nucleolus is present. Look at here. This nucleolus will disintegrate. Here there is formation of microtubule between centriole. Okay, this is prophase, where exactly disintegration of nuclear membrane occurs, disintegration of nucleoli occurs. Okay. So let me remind you again, guys. So you can see here, this is chromatin fiber. Here it is in form of chromatin. Now it gets condensed to form chromosome. So let me recall you guys here. That is prophase. So characteristic feature of prophase. So chromosomal material condense here. So condensation of chromosome occurs. Condensation of chromatin fiber to form. So now you can visualize chromosome distinctly in prophase. So initiation of mitotic spindle. So microtubules are formed. Microtubules are formed. Then next what happens? So here disintegration of nuclear membrane and nucleolus. That means nuclei. Disintegration occurs here. So when you, when we viewed under the microscope, do not show the Golgi complex, ER, nucleolus or nuclear envelope. Disintegration of Golgi and ER also occurs here. So this is what a prophase is. So before that, the chromatin, before that, for example, this is interphase nucleus. So it will be having this type of chromatin fiber. Now in prophase, you will be having, this nuclear membrane will get disintegrate. So let me draw it here. So it will be like this. Disintegration started, no nucleus, nucleolus, and chromosomes you can see now properly. Okay, next it is metaphase. Next is metaphase. So in metaphase, spindle fibers attaches to the kinetochore of the chromosome. All the chromosomes are aligned in the equatorial plane. All the chromosomes align at one plane like this. The center equatorial region. This is forming a metaphase plate. This is called metaphase plate. And all the chromosomes, they are attached to the centromere region with the kinetochores. So spindle fiber attached to kinetochores. Okay. Next, chromosomes are moved in the spindle equator and gets aligned to metaphase plate. So that is done. Next phase is anaphase. Anaphase. What happens in anaphase? Now chromosome arranged at the metaphase plate split simultaneously and into two daughter chromatids. So refer to the chromosome of the future daughter nuclei. So here centromere of each chromosome is moving towards the pole. See, for example, this is the centromere part. Okay, so this gets divided here, and then this chromatid will be like this and like this. Now they move in this direction. Okay, so splitting of centromere occurs. And chromosomes move or chromatids move in opposite direction. And last phase of karyokinesis is telophase. So telophase is just opposite to prophase. It's opposite to prophase. That means all these things which have been disintegrated, the formation of all these things, the reformation of all these things will start. So chromosomes cluster at the opposite spindle poles. They added discrete elements, nuclear envelope, and all these things, Golgi and ER and nucleolus, all these things are reformed again. Then after telophase, it will undergo cytokinesis. That is cytoplasmic division. See, look here. So this is what late prophase. So you can see sister chromatids of each chromosome. They are attached to the spindle fiber. Okay. And centrioles are on the other side. Now metaphase, all of them are arranged at equatorial plate. This is called metaphase plate formation. And then this centromere, it is divided, to split it to form daughter nuclei. Now they move apart from each other in opposite pole. Now in telophase, this is telophase. In telophase, reformation of nuclear envelope, nucleolus will started, and which will lead again next to in case of plant. In case of plant cell, cytokinesis is different as compared to the animal cell. In plants, there is development of cell plate like this, and in that it will grow outward. Okay, because of cell wall presence in the plant cell. But here this furrow will get deepened in this direction, and in turn there will be separation of two cells. I hope this is clear, everyone. Is there any doubt in mitosis? Now, significance of mitosis. Significance of mitosis. So it is an equational division because cell, 2n cell will form 2n cells only. So it is and two daughter cells are identical to the respects. That's why it is known as equational cell division. So they receive the same number and kind of chromosomes as well as in the mother cell. That means for example, the cell it is having 46 chromosomes. That means 2 into 23. Okay, so which will give you two cells by mitosis which has same number of chromosome. Okay, so that's how they are identical, and this is why it is known as equational cell division. So it is the only mode of multiplication in unicellular organism. So unicellular organism multiply by mitosis. So it is the process by which growth takes place in animal and plants. So in ourselves, growth takes place by mitosis only. So somatic cells are formed by mitosis. Somatic cells are formed by mitosis. It maintains the genetic continuity and equality. So this helps in proper coordination among the different cells. So it also plays a role in repair by growth, healing, regeneration of damaged part like in case of tail of the lizard, replacement of the loss during the normal wear and tear. So this as the surface cells of the skin or red blood cells. So this is, now we are talking about meiosis. Now we are talking about meiosis. So meiosis is divided into two phases, meiosis 1 and meiosis 2. Meiosis 1 is also known as reductional cell division, and this is equational cell division. Second, meiosis 1, it is further divided into pro, meta and antelo. But prophase of meiosis 1, that is prophase 1 is divided into 5 different stages. 5 different stages. These 5 different stages, these 5 stages are leptotin, zygotin, pectin, diplotin and dikinesis. So leptotin, it's a stage the chromosome becomes gradually visible under the microscope. So same just like prophase, leptotin. Chromosome becomes visible here. Chromosome becomes visible here, and this is followed by the second stage, the compaction of the chromosome throughout the leptotin. Continuously, it will be forming. It keep on condensing. Then next step is zygotin. Next step is zygotin. Here, pairing or synapsis occurs. Synapsis occurs. That means two homologous chromosomes, they come closer to each other, and to form this is called synapsis, to form a synaptonemal complex, and this is also known as bivalent or tetrad. So in this, which will lead to formation of bivalent or tetrad. That means it will be having 4 sister chromatids. Now next step, it is pectin. Next step is pectin. So in pectin, bivalent chromosome now clearly appears as a tetrad, and recombination nodules are formed. So appearance of the recombination nodules. Okay, so crossing over occurs here. Crossing over occurs here. It is also enzyme mediated crossing over. For this enzyme required, it is recombinase enzyme. Next step that is diplotin. So in diplotin, I'll write it here, otherwise you will get confused. Wait a second. So next step is diplotin. So in diplotin is recognized as dissolution of the synaptonemal complex. That means here chiasmeta formation will be there. See what happens. For example, this is one chromosome. I'll draw other chromosome with other color. Wait a second. Everyone, this is other chromosome. So this type of crossing over, crossing over occurs here in pectitin. And wherever there is crossing over, there is exchange of the genes occurs, and this exchange of genes, then there is still they are attached here. And in next step, what happens in the next step? This is start moving apart from each other. This is start moving apart from each other, but it is combined here at one point. There is one chiasmeta formation. This is one swastik-like formation in this diplotin. Okay, it start moving apart from each other. That is dissolution of synaptonemal complex. This bivalent is getting dissolved now. Now still they remain attached at one point of crossing over. That is stretched out, and that is called chiasmeta. Okay, so X-shaped structure you can find here. This X-shaped structure is called chiasmeta. So in oocytes of some vertebrates, diplotin can last for months or years. In some vertebrates, in oocytes of vertebrates, it can last for months or years. So it will not separate it after too long time. Then final stage, it is dikinases. Next stage, it is dikinases. So this is marked by termination of the chiasmeta. From that, it is getting broken down. Okay, there is exchange of genes occurs, and by the end of the dikinases, nucleolus disappears. So this is end of the prophase. So what could be the characteristic feature of the prophase? Dissolution of the nuclear membrane, disintegration of the nucleoli. So all these things getting disappear. Now disappearing of all these things occur. Okay. So chiasmeta, termination of chiasmeta. Where is my cursor? Termination of chiasmeta. And disintegration of nucleolus, ER, Golgi, etc. Wait a second, guys. So this is about prophase 1. Okay. Next step, it is metaphase, anaphase, telophase. It is same. Metaphase here. What is the difference here? Here bivalent chromosome align at the equatorial plane. Microtubules form the opposite pole and the spindle fiber attaches. So metaphase is same for both the things. But anaphase is different. Because homologous chromosome separates here. There is no splitting of here. Mark this point, guys. In case of anaphase 2, in case of anaphase 2, sorry, anaphase 1, no splitting of centromere. No splitting of centromere occurs. Homologous chromosomes have been paired now, so they are getting separated. Each chromosome is getting separated. There is no sister chromatid separation of the sister chromatid required here, because it is reduction self division. Okay, 46 message 23 it will be. It become 23 now. Okay, then in next step of the meiosis, that is meiosis 2, in that phase, this 23 get replicate and it will get duplicate to form form two new cells that contains 23. That is equational cell division, again same like mitosis. So this is meiosis 2. It is same like mitosis. So, and what is there in mitosis? Prophase, it includes prophase 2. Prophase 2 will be, it is like that. There is a disintegration of nuclear membrane, disintegration of all the nucleus, ER, Golgi apparatus occurs, and you can visualize they get condensed, chromatin gets condensed. Okay. So here prophase 2. So mitosis 2 is initiated immediately after cytokinesis. So in between there is no cytokinesis. Okay, between cytokinesis is not there here. After telophase 1, it is known as interkinesis. Between phase is known as interkinesis. Okay, it is starting immediately. So sometimes you can say prophase, the nuclear membrane get disappeared by the end of the prophase 2. Then metaphase, they again align at the equatorial plane. Anaphase, then they move apart from each other. Now splitting of centromere occurs. Look here, this is leptotene. This is your prophase 1. So prophase 1. So leptotene, diplotene, dikinesis, leptotene, zygotene, diplotene, pectine, then diplotene, then dikinesis. Okay, then it will lead to metaphase 1. You can see here how metaphase 1, all the chromosomes, they are at equatorial region. So each chromosomes are moving apart from each other in anaphase 1. There is no splitting of centromere. Then which lead to dikinesis and prophase 2. It enters both the cells enters into prophase 2. Then metaphase 2 and anaphase 2. Now splitting occurs here. Here splitting occurs, which will lead to formation of 4 daughter cells. I hope this much is clear about cell division, about meiosis also. Second stage, it is second meiosis part 2. It is same as mitosis. Then significance of meiosis. Meiosis maintains the chromosome number from generation to generation. Yes, Bhavna, hello, very good evening. Those who have joined, they can wave your hand in chat section so I can know your name. I can call out your names. Meiosis maintains the chromosome number from generation after generation. Otherwise, during fusion, during fertilization, chromosome number gets doubled every time. If it is not getting reduced, then it is getting doubled every time. So it reduces the chromosome number to the half so that the process of fertilization restores the original number of the zygote. Meiosis occurs during the gamete formation and reduces the number of chromosomes from diploid to the haploid. So gametes contain haploid chromosomes. These haploid gametes fuse to form zygote. Variations are caused by the crossing over and the random distribution of the homologous chromosome between daughter cells. So variations, the sexual reproduction is important for variation because sexual reproduction involves meiosis, not mitosis. So it's very important. It plays very important role in evolution. Chromosomal mutations are brought about by the introduction of the certain abnormalities, and these chromosomal mutation may be advantageous for the individual. So these are significance. You can look into the difference of the mitosis and meiosis. So in mitosis, single division result into 2 daughter cell. Meiosis require 1 and 2, which will form 4 daughter cell. Mitosis known as equational division. This is reductional division. Prophase is short here in mitosis. Not comprise any different phases. In prophase 1 of meiosis, it has 5 different phases. There is no pairing of the chromosome crossing over chiasma formation in mitosis. All these things occurs in prophase 1 of meiosis. Synaptonemal complex is not formed. Here it is formed. Anaphase involves the separation of chromatids. See, and keep on revising that thing, keep on recalling that here. This chromosome, chromatids of each chromosome is getting separated by splitting of centromere. Here during anaphase 1, no splitting occurs. Okay, chromatids remain attached to the centromere, but in 2, they get separated. So mitosis plays a significant role in healing, repair, growth of the cell. Meiosis, it is required for variations and maintaining the chromosome number from generation to generation. So again, I am reminding you, all of you, you can download the app for great discounts on our courses. You can apply the code, that is VAI. Those who have already joined, they can pass this message in their group, and those who want it right now for the crash course, they can apply this code, that is VAI 10, that is 10, VAI 10, that is 10, to get great discounts on all our courses. And you will be, then when you apply, when you use this code, this coupon code, you will be enlisted in my student list, and we will be directly connected by then. Okay, so after that you will be having handwritten notes, study materials, PYQs, mock tests available in that. So this is our Spark store. All these things are available right now. Links are given in the description box, so you can purchase any of these things from this. This is how classroom coaching, so it's open right now. Join us right now for crash course, which have been started on 18th April. So it has been only 1 week, still it's on the starting part. So you can join right now. Do not, this is high time you can join the offline crash course. Okay, online crash course will be starting soon. You can join any of the branch, Narayangura or Mehdipatnam. Online coaching is also there, and for that also you can use the same coupon code, that is VAI 10. On daily basis we'll be having live sessions. We will be having live sessions, and this is how we are giving assignments on daily basis. We are creating that assignment on our app, so you can check it down whether they have submitted it or not, or pending or like that. So exams are conducting every weekend test and grand test, and we are doing test analysis also. This is our method. And why only Spark? Because we have highly experienced faculty, and we believe in personalized coaching, and we have very limited size. We give them personal mentoring and doubt clarification. So this is how, if you opt for hybrid coaching, you can have access to online coaching also. But when you are opting for the online one, you will be not having access to the classroom. Okay. So first question answer time, guys. Which of the following is not a characteristic feature during the mitosis in the somatic cell? Come on. Quickly. Come on, I'm waiting for your answer. Those who have joined, we are lacking time. Come on, make it fast. So, not a characteristic feature during the mitosis. So not, you need to focus on a not word. So spindle fiber formation occurs, disappearance of nucleus also occurs, chromosome movement occurs, synapses do not occur in mitosis. So this is a correct option. The complex formed by a pair of synapse homologous chromosome is called complex form is known as bivalent or tetrad. During metaphase stage of mitosis, spindle fiber attaches to the chromosome at kinetochores. Kinetochores, they are disc-like structure at the centromere region. Centromere, kinetochore, and areas adjoining the centromere. So they attach at the kinetochore. During meiosis, one chromosome starts pairing at. So pairing is started in zygote. Pairing is started in zygote. Synaptonemal complex formation. Synapsis started in zygote. Chromosome in which centromere is situated close to the one end are called, two are short arms and two are long arms. So these type of chromosome is known as acrocentric. During which phases of the cell cycle amount of DNA in the cell remains at 4C level and its initial amount is denoted as 2C. So in which phase it gets double? See, we have discussed this during the discussion. So in S phase it gets double. So S after that G2. So in S it gets double. So after S it's only in G2 phase. It will remain 4C. S phase of the cell cycle, the amount of DNA in each cell is getting double. Enzyme recombinase is required at which stage of the meiosis? Enzyme recombinase required in which stage of the meiosis? That is SPARKACADEMY. Somatic cell that has just completed the S phase of the cell cycle as compared to the gamete of the same species has same number of chromosomes but the twice amount of the DNA when it is compared with the gametes. Okay. Somatic cell, see I am explaining you here somatic cell, that means 2N. Okay. And when you're comparing it with gametes, this is gametes. So gametes, they have same number of chromosomes. Okay. But twice amount of DNA. See, somatic cell that has just completed S phase. S phase has been completed. S phase has been completed. S phase has been completed here. That means amount of DNA, it is 4C here. Number of chromosomes 2N. Of its cell cycle compared to the gametes. So gametes of the same species. Gametes have N and it contains 1C only. So same number of chromosomes but twice amount of DNA. No. Twice the number of chromosomes and 4 times the amount of DNA. 4 times the number of chromosomes and twice the amount of DNA. This one is correct here. I hope you are getting this point. Next, DNA replication in bacteria occurs where exactly? It is occurring in cytoplasm prior to the fission. Fission that means division into two cells. So before fission, it occurs in the cytoplasm. Again, let me remind you, all of you, you can join us on Telegram also, that is Spark Academy. Our link is here. Now these are your homework questions, so quickly take a screenshot and I'll be waiting for your answers. Next question, correct option for with respect to mitosis. Next question, at what stage the cell cycle are histone proteins synthesized in the eukaryotic cells? Sorry, again I am repeating the question. So at what stage the cell cycle and histone proteins are synthesized? Next, select the correct option. Then metaphase, chromosomes are attached to the spindle fiber by their satellites, constriction, kinetochores or centromere. Don't forget to subscribe to our channel, press the bell icon for notification. Don't forget to send this video, share this video to more people, and don't forget to press the thumb icon, that is for the liking this video. I'll be waiting for your likes and I'll be waiting for your comments in comment section. Thank you so much. This is our EAMCET result 2021, NEET 2021. Thank you so much. Don't forget to like, share and comment in comment section.